So thank you all for coming. Our first speaker is Stuart Lichtenberg, and he is going to be talking to us today about his nanotechnology project and nematodes.
Hello, everybody. Thanks for coming. Like I said, I'm going to be talking a little bit today about my project, which mostly focuses on nanotechnology, RNA interference, and the combination of those two as next-generation pesticides. So first, a little bit of background, sort of explain what exactly we're working with here. So RNA interference is basically a process by which we can selectively silence genes. So shown here is the central dogma of molecular biology. That is to say, DNA makes RNA, which makes protein. Well, in most eukaryotes, we can introduce an artificial double-stranded RNA molecule as a template to serve for the degradation of a given RNA. When this RNA is degraded, it obviously can't be translated, which means you don't get a given protein. Now, this process has been utilized extensively for functional genomics for many years now, but it also has the possibility to serve as a pesticide. If we target, for instance, an essential gene in an insect, we can use it to selectively kill that insect. Currently, there's a number of products that are about to hit the market that utilize RNAi-based technologies, but these are all exclusively based upon transgenic products and only target a limited number of species, such as corn rootworm. There's also some serious caveats with using double-stranded RNA or RNA interference products as a more of an in vitro solution, in that major double-stranded RNA transporters are absent in some crops. Some pest species, general cellular uptake mechanisms are slow or selective for molecules that aren't necessarily double-stranded RNA. And furthermore, there's more of a general concern that double-stranded RNA is very rapidly degraded in the environment. Studies have shown that double-stranded RNA persists for less than 24 hours in natural soils. So with these problems in mind, what are some of the ways we can get around these? One such way is the development of things known as polyplex nanoparticles. Basically, in these systems, what we do is we combine double-stranded RNA, which is negatively charged, with a positively charged cationic polymer. These will then condense into small nanoscale objects, which can be taken into cells. So to really have agricultural utility, we need a cationic polymer that's cheap, abundant, and generally regarded as safe. The one that we're going to be looking at today is known as chitosan. Chitosan is made from shrimp shells. Basically, you just take shrimp shells and boil them in sodium hydroxide and get chitosan. So it's abundantly available. It's also generally regarded as non-toxic in most studies. So our working hypothesis here was, if we make these chitosan double-stranded RNA nanocomposites, would they show increased efficacy in a model system of some kind? And for a model system, we chose to look at the efficacy of these particles in C. elegans. C. elegans is kind of the ideal model organism for a whole lot of different things, but in particular looking at RNAi. RNAi was first characterized in C. elegans. The transport and uptake mechanisms of double-stranded RNA are very well established in C. elegans. It's also easy and quick to generate stocks of these organisms as well. So the figure at the right sort of shows the general uptake mechanism of double-stranded RNA in C. elegans. Essentially, an organism will ingest double-stranded RNA, and a series of receptors and transporters known as the SID proteins, SID2 and SID1 in particular, take the double-stranded RNA from cells lining the gut and further propagate it into cells throughout the body. So then we need to get an actual model system to detect whether or not our particles are effective. And in this case, we used a particular strain, a particular transgenic strain of C. elegans known as CGC4. CGC4 has GFP driven by a promoter that is for a translation elongation factor. So it's abundantly expressed in pretty much all cells in the worm's body. And we can see that in the figure at left. These are control worms that haven't been exposed to any chemicals at all. We see bright fluorescence throughout. And on the right is an image of a worm that's been exposed to double-stranded RNA. We can see there's a distinct reduction in fluorescence in this one. So basically, to see if our particles are working, all we have to do is expose the worms to them and see how much they're glowing. Then the first question was, is there a difference between chitosane nanoparticles versus naked double-stranded A on a concentration, on an exposure concentration basis? And generally what we found is there actually isn't much of a difference. We can see this here in the graph. There's a little bit of differentiation at a few places, but by and large, there's not a terrible lot of difference. However, we did find some interesting differences when we started to look at the actual amount of double-stranded RNA that's internalized by these worms, depending on the form in which it's presented. So we had a few different exposures here. The top left panel here is chitosane that we've labeled with a fluorescent marker. We can see there's a relatively small amount that's internalized into the worm. The panel to the right of that shows this same labeled chitosane, but put into a nanoparticle form. So we've added double-stranded RNA. Similar results, not a terrible lot of it is internalized. Now the bottom left panel here shows double-stranded RNA alone that we've labeled with a yellow fluorescent marker. We can see the worm takes up a pretty significant amount of double-stranded RNA here, coats the entire gut, and has bright fluorescence throughout. The last panel there shows dual labeled particles, ones where we've labeled both the double-stranded RNA and the chitosane. We can see a similar distribution of both and some co-localization as well throughout the worm. And when we actually measured the intensity of the fluorescence of double-stranded RNA in just double-stranded RNA exposed worms versus nanoparticle exposed worms, we did indeed find there was significantly more uptake of double-stranded RNA when presented in a naked form than with a nanoparticle, showing that pretty much, so even though the nanoparticles themselves were similarly effective on a concentration dose basis, in terms of a tissue concentration basis in the worms, the chitosane particles appear to be quite a bit more effective. That then led us to the next question, which is, why is that? Is it because these nanoparticles are protecting the double-stranded RNA degradation? Is it because there's a different uptake mechanism? And we decided to approach that question next, in that we wanted to look at, well, how exactly is the double-stranded RNA taken in nanoparticle form? So we looked at a few different null mutants for the transporter genes that I mentioned earlier, SID1 and SID2. These are strains that normally, if you expose them to double-stranded RNA through environmental exposure, through taking it in through their gut, RNAi is completely ineffective. It doesn't do anything. So we had our double-stranded RNA in our nanoparticles, and we can see in wild type, we see gene knockdown in both cases. Both of our treatments are effective. In the SID1 and SID2 mutants, we can see double-stranded RNA has no effect on gene expression. There's no significant difference. However, when we present the double-stranded RNA as nanoparticles, we do see a significant reduction in this reporter gene. So effectively, our nanoparticles are somehow evading the traditionally understood uptake mechanisms of double-stranded RNA in these worms. Then the question is, if it's getting around these, how exactly is the double-stranded RNA getting into the cells? Our attention then turned to the possibility that it was clathrin-mediated endocytosis, sort of a more general means that the cell uses to take in materials. So to address this, we exposed our worms as the same as before, using chitosane and double-stranded RNA, but we also included a substance known as chlorpromazine in the exposure as well. Chlorpromazine is an inhibitor of clathrin-mediated endocytosis, and we can see In both cases, this inhibits uptake of double-stranded RNA. We don't see any difference in gene expression when we introduce chlorpromazine. This was to some extent expected with the double-stranded RNA because it's known that the SID system is closely tied to endocytosis and lends evidence to the idea that it is in fact platinum-mediated endocytosis that's bringing these particles into cells. Oh, and just a quick aside, chlorpromazine is also more popularly known as Thorazine. So if anybody does start tripping from the chili this afternoon, like Jason continued with the Simpsons picture, I got the hookup and I can make the trip stop. That's not the end of the story, though. Going back, I mentioned that we were using a strain known as CGC4 with a particular promoter that drives GFP expression. This was not the first reporter strain that we had used. In fact, the first reporter strain we were introducing was known as PD4251, which has GFP driven by a myosin promoter. And we found something kind of interesting in this particular strain. So when we expose this particular strain to GFP double-stranded RNA, we see a reduction in fluorescence, as we would expect. When we have chitosane nanoparticles with GFP double-stranded RNA, we also see a reduction in fluorescence, which we would expect. When we have a scrambled GFP as a negative control, so this is a GFP double-stranded RNA that has no sequence homology to actual GFP, we don't see a reduction in fluorescence. Again, what we would expect. However, when we present it in the form of chitosane nanoparticles, even with the scrambled sequence, we see a large reduction in fluorescence in these worms. So what's going on here? We couldn't find any reasonable explanation as to why a scrambled sequence would be affecting GFP expressions. The only thing we could think of is that in some way, chitosane was affecting myosin expression, since it's a myosin promoter driving expression of GFP in this case. So we were able to go and confirm that with QRT-PCR and found that, in fact, chitosane does suppress myosin expression. This has some pretty significant implications because myosin is ubiquitous in eukaryotic cells. It's required for a number of cellular processes, including growth and reproduction, and it's heavily involved in muscle contraction as well. So we wanted to see if there was any possible effects associated with this reduction in myosin levels. And consulting our data sets previously, we did find that there was a significant decrease in body length for worms that were exposed to chitosane nanoparticles. And even at really low levels of chitosane exposure, we saw significant reductions in fecundity as well. So even though chitosane has been generally regarded as safe and non-toxic, we have found some sublethal effects that are relevant in this case. Sort of illustrates both the need for proper assessment of these materials prior to field scale introduction. So in conclusion, we found that chitosane nanoparticles don't dramatically increase dsRNA efficacy on a concentration dose basis, but do on a tissue dose basis. They enter cells via platinum-mediated endocytosis, not the traditionally understood means of double-stranded RNA uptake. And they also reduce expression of myosin and C. elegans, leading to sublethal effects that were previously uncharacterized. With that, I'd like to thank my committee and the folks in the Environmental Chemistry and Toxicology Lab, and I'm open to any questions.
One of the hallmarks of organic agriculture is growing all sorts of tightly containing waste in the soil inside of control. What then is the implication of that particular practice on hematose population?
Well, actually, so it's hard to say. So I should point out that chitosane is what we might call semi-synthetic. It's not very abundant naturally. So to get chitosane, you have to take chitin and, like I said, boil it in sodium hydroxide for a period of time to deacetylate those amine groups. There could be some implications from nematode control. Like I said, we've obviously seen some sublethal effects here that haven't been characterized before. So maybe, hard to say.
You want to see if Princeton has any questions?
Princeton, do you have any questions? Okay.
All right, let's thank our speaker one more time. And our next speaker is Elisane Tessmann, and she will be talking to us about her leaf reading project.
Thank you, Catherine. Thank you, everybody, for being here. I'm very excited to share with you guys a little bit of my research. So this study was actually published in the beginning of the year in the Agronomy Journal. And what I want to do now is, this will be the outline of the presentation. So we have a brief introduction, objectives, materials, methods, some results, and conclusion. So what I want to do is to break down the title in three portions and explain the idea of the experiment. So the first part will be about disease, Fusarium hemorrhoids. So this disease is caused by Fusarium miniarum. It happens during the flowering in wheat, and it needs warm temperature and high humidity. So as you can see here in the picture, some of the symptoms that we see in the plant when we are in the field. So we have leaf spikes, and we have damage in the grains. We're going to reduce yield, and most important is mycotoxins such as the oxynivalenol or DORN. They are very toxic when we consume that grape. So why does that matter? We do not have resistant cultivars because we need multiple genes to compare resistance to this disease. So the second portion is about climate change, and that goes to our type of experiment here. It was the artificially warm treatment. So it is predicted that we're going to have an increase from 1 to 3.7 degrees Celsius by the end of the century. So just to illustrate in pictures, if we look back to 1967, we can see that the Earth was a cooler environment. So blue means there was colder, and then when you go to orange and red, they were more warm. So here how it was in 2000, and let's see in 2017. So you can see that we have a drastic, more warm environment compared to 50 years ago. So, and with that, we're going to have changes in rain. It is predicted that if each increase in degrees Celsius, we're going to have a very high reduction in yield production, and it's going to affect the distribution of disease and intensity, but we don't know how they're going to affect. And the third portion of the experiment title is about the genome-wide association study. So in this portion, we use a large set of cultivars and plants, and we phenotype them in the field. We have genotypic evaluation, and combine both using statistics models. We can then identify PTLs or SNPs that are going to be related to specific traits that we are evaluating. So that the objective of this study was to evaluate phenotypic response to FHB and other disease traits in a diverse wheat panel under warming control conditions, and then why did I go back here? And then determine whether we were PTL associated with FHB traits under warm conditions when compared with control. So the experiment was conducted here in Langston in the Spindletop farm. We used 238 cultivars that came from different breeding programs across the U.S. We planted in 2 years, 2015 and 16, in an experimental split plot design. Due to the number of cultivars that we have, we use few plots, and we have 2 reps in 2016 and 4 reps in 2017 under warming control conditions. So here in the top, just to show how the experiment looks, we have 6 rows, and we have in between the rows, we have the cables that are buried in the soil, and then going to warm the rhizosphere. The blue cables, they measure soil temperature. So we have cables in the warm treatment, and we have the cables in the control. They're going to measure the soil. So the cables were turned on every 3 minutes. If the difference between the environment and the soil was more than 3, then the cables were turned on. And every 15 minutes was reported the temperature in the soil. So to evaluate the disease, we spread cognitive solutions in 2016. And in 2017, since we didn't have another experiment around, I could actually use Capricorn and provide a more efficient infection. So we also use the overhead irrigation system to have more disease in this field. So the traits that I evaluate, heading date, plant height, FHB rating, there was is a visual disease, note of disease that goes from 1 to 9. 1 meaning 10%, 9 meaning 90% of disease, and yield. And then in 2017, because we had a more severe disease level, I was able to actually evaluate FDK, that is Fusarium damaged kernel, that you can show the picture here, that goes from 5 to 9% of the kernels, they are damaged. So it was a visual scale that we gave. And the oxyvalenol DON, that was the toxin that the fungus produced in the grain. So we send our samples to another university, and they do the DON analysis for us. And then other visual also disease that that I saw in the field. So the analysis I did ANOVA, the heritability correlations I'm not presenting here because I don't have time to show all the data. And then the GYS. So the data was sequenced in Ohio using the 9K Illumina chipset. Around almost 4,000 SNPs were used. And since we have information for important details, I use those the covariance in the model. And you use the problem to do the math. So here I want to show first. So I had cables in the warm and the control treatment. So here shows the difference in temperature from the warm and then in control in 2016 and 2017. So you can see that the cables were efficient in keep the difference between warm and control. So every time that you see an orange color, they're going to be referring to the warm treatment, and blue is going to be the control throughout the whole presentation. So here there's another table. We can see that we have different, all the genotypes, all the traits that we evaluate, they were significant different. And from the treatments, there was the warm control, the genotype. We have difference from the disease phase that we evaluate in the view. So to show a little bit of how the genotypes did in each year. So this is a mean comparison between warm and control. So for heading date, so we have the days here and the growing season, 2016 and 17. So we see that in general, in average, independent of the treatment, 2017 was 8.4 days early in the season than in 2016. And if you look in the specific year, the warm treatment was efficient in reducing the heading days from 3.5 in 2016 to 3.4 in 2017. For the disease, since 2016 we didn't have a lot of disease that we don't see difference. We didn't see difference in the data, but since 2017 was more intense, we had more disease in the field. We were able to actually see a little bit of difference in disease. For DON and FDK that were evaluated in 2017, we see that DON, again, the warm was very efficient in the cause disease in the plant. So we see an increase of 84% in DON and 131% in FDK compared with the control. So this is our GYS analysis, how we sum the data from when you run the analysis. So in total, we identified 29 SNPs, where 19 were in the control treatment and 10 were in the warm worm treatment. And I want to focus more on the FDK and DOM because those are the disease that we were evaluating, disease phase. So here in the control, we identified 7 diseases, where 3 had a negative effect, that is what we actually want, because we want to select something that have a negative effect or reduce our disease. We didn't see anything in the worm treatment. And for DON, in the control, we identified 5, where 2 had a negative effect. And we did not, and 3 needs on the worm treatment, but they were not negative. They didn't have a negative effect. And then since we have the KTL information, I also want to show how the means for each genotype in each one of those important KTLs. So FXB1 is a gene that compares resistance or some sort of resistance to the plant. So we have 19 plants that have that gene of our total of 238, and we see that there is no difference. There is no difference between those plants. And the genotypes that didn't have that disease, then KTL, then we see a more difference in the worm, where we have the worm causing more disease than in the control treatment. And that trend happened for all the other KTLs. For example, for the high KTL, so the increase was 69%. For the reduced high alleles, we see that the worm treatment reduced 50 and 78%, increased 50 and 78%. We have more disease on the worm treatment. And then similar here for photoperiod, we also see an increase in DON levels on the worm condition. So overall, the worm treatment, it was more efficient or caused more disease than plants in the control. So the take-home point of this experiment was that 2 degrees Celsius increase in temperature resulted in an early hanging date of approximately 2.5 days or 4 days. The disease levels were higher in the worm treatment than in control treatment. The disease identified in the study can be useful for selection. There's a great difference between worm and control treatment for all the known and important KTLs in which, independent of the allelic form, the worm was always caused more disease than in the control treatment. And with that, it's necessary more screening under worm conditions to evaluate our profile. And I just want to say thank you to this CAP initiative. It's the finance my project, the Wheat Breeding Program, my advisor, the Elisane Tessmann, John, Sandy, Catherine, Jess, and Virginia for all their help in the field. And a special thank you to Martine and Brian for helping me digging more than 2,000 holes. And then later on in the season, when it was really cold and the soil was wet, digging holes so we could put the cables in between the plants. So thank you so much.
Any questions for our speaker?
That's it. And then record for the increase in temperature from 1 to 3.7. Is that based on air temperature or soil temperature?
Air. I think it's air.
So do you think it will be quite as dramatic in terms of disease presence, yet the soil temperatures tend to be buffered with respect to temperature?
Well, my experience was more in the soil, so I was warming in high stratosphere, so it was 2 degrees. So I think if the environment was going to be also, between 1 and 3, I think the tendency is to get worse than 1 and 5, because by warming the high stratosphere, I changed the phenology, right? So my plants were earlier. So if you also have an environment, a warm environment, I think you potentially can have more disease. Of course, you need to have a humid environment, so you also need to have rain to have the disease. So if you have an increase in temperature, but then you don't have rain, so you're not gonna have disease. So it's hard to know what's gonna happen. I don't know if I answered your question.
I had a similar question.
Okay.
All right, let's thank our speaker. Our next speaker is Foteh Ali, and he will be talking to us about actin and regulators in Arabidopsis.
Welcome everyone. Before I start my presentation, I would like to ask one question. Can you imagine your single daily meal without contribution of seeds? Probably not. Yes, seed is very important not only for plants for its reproduction, but it's also very important for humankind for food and for livestock feeding. Our lab is mainly focused on seed molecular biology, and especially my project is mainly focused on actin and its regulators in stenocytic endosperm development that I will explain later to determine the seed size potential in Arabidopsis thaliana. In most of the angiosperm, specifically in Arabidopsis, seed development actually contains pollen tube contains two sperm cells. One of them fertilized with egg cell to generate a nice embryo, and another one fertilized with central cell to generate the endosperm. And endosperm development has several stages, and the initial stage or early stage that we call the stenocytic endosperm development. And this stenocytic endosperm development is the stage when endosperm enlarges very rapidly, and the endosperm nuclei, here you can see the endosperm nuclei, those are divided without any cytokinesis. And after rounds of nuclear division, the endosperm starts to cellularize from micropylar end, and when the embryo actually reaches a torpedo stage, the endosperm completely cellularized. From several research work, we know that the transition from the stenocytic endosperm to cellularization is very crucial, and that's linked to smaller or bigger seeds. So here I put one example where the wild type Arabidopsis plant showing the endosperm cellularization. Here you can see this is 24 hours after after pollination. This is 36 hours after, and this is 48 hours after pollination. And in this picture, you can clearly see like there is no endosperm cellularization happening here and the tiny embryo. But when they check the endosperm cellularization in AGL62 mutant, and you can see at 36 days after pollination, the endosperm here, you can see the endosperm is cellularized. And after that, at 48 hours, you can already see the seeds is smaller. So this mutant is actually ultimately giving the smaller seeds. And not only genetics, in my master thesis, I used two interspecies of Arabidopsis arenosa and Arabidopsis lirata, and I checked the hybrids on the basis of endosperm development. And here you can see this is the 9 DAP Arabidopsis arenosa seeds, this is 50, and this is a 9 DAP Arabidopsis lirata, and this is 15 DAP. And when I crossed Arabidopsis arenosa as a mother with lirata, here you can see in this picture endosperm already cellularized. And these seeds, this image actually represents these smaller seeds. On the other hand, when I crossed the arenosa Arabidopsis lirata as a mother with arenosa, here you can see at 15 DAP, all seeds are developed, but still it's not cellularized. And this image actually represents this aborted blackish seeds. So these interspecies in cross is actually somehow controlled by epigenetic mechanism. So from this experiment and previous experiment, now we know that from epigenetic, there is a tightly association between the stenocytic endosperm to cellularization, which is actually showing smaller or aborted bigger seeds. So now we know that this transition is very important, but before this transition, the stenocytic endosperm is also very important because there are so many interesting things are happening. And for my project, I'm very interested to know like the role of the synocytic endosperm to determine the seed size. And to understand the synocytic development, recently in our lab, we have developed the live cell imaging to capture the entire synocytic phase of endosperm. And here you can see the nice image, and Dr. Tomo actually used the endosperm specific FWA promoter to activate live venous to visualize the egg active. And here you can see the green cables are egg active, and the same thing the FWA promoter to with the H2B nuclei marker to mark nuclei. And here you can see the magenta color, those are marking nuclei. So from this snapshot, you can see that F-actin is generating nice ester-shaped structure in the synocytic endosperm, and this ester-shaped structure and F-actin is actually maintaining the nuclear movement in synocytic endosperm. And from my live cell imaging here, I take a video that I want to share with you. Oops. Yeah, from this video, you can see the nice actin actually generating the ester-shaped structure. The nuclei are moving, and we also found that the nuclei in this synocytic endosperm somehow pulling or pushing towards the chalazial endosperm end. We call this part, and they're generating kind of cyst. So from my live cell imaging, we know that there is a actin in synocytic endosperm doing really nice work, and we don't know their details function. So to know the F-actin, we wanted to check some regulators, and there are so many regulators actually regulates the F-actin glands, especially propylene, pillin, formin. And formin is one of the important factors in plants that actually generate actin nucleation and elongation in plants. So formin has actually two domains we call FH1 and FH2 domain. And here, this tail domain, a tail like figure is the FH1 domain, and this green portion is FH2 domain. So this formin 2 domain is actually generate dimer, and after generating the dimer, it starts actin elongation, and after that it starts like nucleation elongation or for this for actin in plants. So after that, we actually checked some publicly available data focusing on the synocytic endosperm, and from the publicly available data, we found few interesting genes. Those are specifically expressed in synocytic endosperm. And from those genes, we identified one formin gene called formin 12 that is also specifically expressed in synocytic endosperm. So after analyzing those publicly available data, I generated the transgenic line harboring the promoter of this formin 12 mutant with H2B to mark the nuclei. And here is the seed after 2 days after pollination. And if you can see, this is actually the synocytic stage of endosperm. And you can see the formin 12 is actually expressing in the nuclei in this stage. I also obtained the single mutant of this formin mutant, and after obtaining the single mutant, first I was very interested to check the seed size of this mutant. And interestingly, when I got the result here, you can see this is the seed size in millimeter square. This is wild type seeds, and in formin mutant, they are even bigger seeds compared to these wild type seeds. So after getting this interesting result, I was very curious, like why this formin mutant giving bigger seeds? And to address this one, at first I checked the embryo development of this mutant, and I checked like 2-4 day after pollination embryos, and here this graph is showing the percentage of embryo, and like embryo has several developmental stages, and I divided like in different categories, like no embryo but the seed is fertilized. After the pre-globular, globular, transition from globular to pre-heart and heart. Here you can see this is 2D8P formin mutant, where I already found pre-globular, mostly all of them. But in wild type, no embryo, but it's fertilized, few of them has pre-globular. In 4-DAP, you can see in mutant already I found hard stage embryo develop seeds, but in wild type no. So from this result, it's telling that the embryo development is actually accelerated in this forming mutant background. After that, I was also curious to check the cellularization of these seeds. And after checking the cellularization, actually, I didn't see any difference between this wild type and mutant background. And as I previously mentioned, that F-actin has an important role in synocytic development, synocytic endosperm development, and this formin 12 is one of the important regulators. So that's why now I want to know, like, in absence of this formin 12 mutant, what's actually happening in this synocytic endosperm. Therefore, I have the mutant. Now I already integrated this formin 12 mutant into the F-actin, and after that, my plan is to check the entire synocytic development in time-lapse image in real time. So from this image analysis, I would like to check like the movement of endosperm nuclei, like in absence of this formin mutant, actually how the movement of endosperm nuclei is changing, whether there is any change of timing of nuclear division or endosperm cellularization or F-actin organization. There are actually so many questions I can address from my next experiment. At the end, in summary, as I showed, like F-actin has an important role in synocytic endosperm development, and we also found the formin 12 mutant, the formin 12 gene that is specifically expressed in synocytic endosperm. So from this preliminary result, now we can think further, like the real-time image that I'm using, it could be one of the important to understand the total process of entire synocytic endosperm development, and further maybe this role of F-actin and its regulators, and how this F-actin and its regulators actually giving the bigger size seeds in terms of like seed development. So those questions will be very interested to understand further. And finally, I'd like to thank Dr. Tomo as my supervisor and other lab members. And now I'm happy to take questions. Yeah.
So you said that your formin was expressed specifically in the endosperm, but you're seeing an effect in the embryo in the mutant?
Yeah, yes.
You want to explain?
So as we know, like it's specifically developed, expressed in synocytic endosperm, but we don't know like maybe some other important factors that are related with the F-actin. So we don't know like whether how they interacting with the F-actin and the F-actin and the formin. So maybe due to this absence of this gene, somehow F-actin is regulating something else that's accelerating the embryo development.
So you confirmed it's not in the embryo?
Yeah, it's not in embryo. Yeah.
You said there was a difference in seed size, but it didn't seem to be very large. Is that based upon a statistical comparison, or is that just the measure?
Yeah, it's statistically like the seed size was bigger statistically compared to the wild type, not just the size. And this is just my preliminary result, and I'm taking more batches of seeds to confirm that, yeah, really I'm getting bigger seeds in mutant.
Right. Thank you, speaker.
Thank you.
All right, our our next speaker is Joshua Knight, and he will be talking to us about irrigation water.
Everybody hear me all right? Okay. Good morning again. Like you said, I'm Joshua Knight. I'm a senior extension associate for the Center for Crop Diversification over in the Department of Horticulture, and this is my master's kind of non-thesis research. It's a summary of unpublished research on some multi-year kind of USDA investigation into water use and water recycling. So I'm going to start kind of defining these terms and then go into some case studies. So we're looking at irrigation water applied, consumptive water use, water footprint, and then the way water breaks down into these different categories of green, blue, and gray water. So irrigation water applied is pretty straightforward. This is the volume of water that comes out of your irrigation equipment. Of course, what we've been investigating here is nursery crop systems. So this was container production, so there wasn't really any field production going on. Yeah, so it makes it a little bit easier when dealing with some of these things. So, like I said, irrigation water applied is what is coming out of your irrigation system for your production. And then consumptive water use is an attempt to measure water based on its impact to other water users, with the main principle being that a consumptive use of water is when you are withholding it from flowing downstream and being available to others. And then the distinction between consumptive water use and water footprint is the water footprint is a weighted version of consumptive water use, and that is weighted by the availability of water both at the time that you're using it or withholding it, as well as the geography, so the watershed that you find yourself located in or withdrawing water from. And so then green, blue, and gray. Green, we'll go into more detail about these, but green water is, it kind of characterizes your reliance on precipitation patterns. So it's water that's falling when you need water in your production system. Blue water, it's going to go out of order. Blue water is kind of your reliance on water stores or water withholding, groundwater, surface water, stuff like that. And then gray water is kind of used to characterize the pollution of your system. And so, kind of articulate specifically on green water, yes, it's rainfall that's being used directly during that rainfall event. Rainfall that hits the water and runs off, that's something else. If the rainfall is hitting it right when you need it, the way we measured it or modeled it was to look at the amount of irrigation events that they avoided due to precipitation patterns. And then for looking at blue and gray water, blue water is going to be any captured rainfall runoff, any additions from other sources like wells or municipal sources or something like that. And then gray water is the volume of water that's necessary to dilute whatever your discharge is to the level that it's kind of arbitrary. It's dependent on where you are and what the water quality standards are. If they want your water discharges to be brought up to drinking level standards, then that is the pollution requirement. It's kind of like, what is your discharge? What is the volume that it's kind of requiring of what you're discharging into? And from the difficult piece of that was kind of modeling captured rainfall runoff. This is kind of the dogma that we operated with to model that. I won't really get into it because I don't have much time. This is normally like a 40-minute talk, but there's some GIS tools for spatial analysis and things like that to delineate watersheds and look where water flows and going from there, and then bringing into the soil types and the hydrologic soil groups, how much runs off and rainfall and cover and things like that. So the systems that we look at in these case studies, a lot of them are essentially artificial small watersheds because they are recycling our water. That's what the water research was about, was looking at water recycling and treatment technologies. So when you weight water footprint, the current standard since last year is this AWARE method. It's supposed to refer to kind of available water remaining is what that stands for. And it's a collection of indexes that are applied on a monthly basis to watersheds around the planet. And so if you have a water scarcity index of 1, that's saying that that watershed at that moment in time has the global average for water scarcity. Small numbers are going to represent water abundance, and then very high numbers represent water scarcity in this kind of comparative system. Prior to that, they tried to do, or were doing, kind of this objective measurement of the available water remaining, or not the water remaining, but the amount of water withdrawn divided by the total water available. But anyway, this is a moving target, and these numbers change on kind of a yearly basis based on how much water is being extracted and things like that. So we'll kind of get into the case studies here. One of the first place that we looked at was this nursery in the Mid-Atlantic. Fairly large, not the largest that we looked at. I mean, their watershed area was almost 500 acres, but then their production area was, over 100 acres. And they had a couple of large ponds that are kind of highlighted there. And this is kind of what the characterization of their water management looks like using those kind of different terms. And down here, we've got sort of this climate sort of watershed characterization of the scarcity indexes that you see, or indices throughout the year, and then the rainfall of 30-year averages. And you can kind of see, they're recycling their water. They have it set up so they're irrigating, and it all flows back into a treatment pond, or it flows back into a reservoir, and then it's treated and applied again and again. So this blue water, this consumptive use and blue water footprint is kind of a way to figure out how much water you're really using in these systems where you're recycling. And so, most of that is going to be due to them capturing rainfall runoff in that system as well, not allowing it to flow down the stream. And there's a slide at the end where we look at all these side by side. The next case study was one in the Pacific Northwest, very large, 500 acres of production space, a much flatter area, and you can see it's not really contiguous, so it's kind of these two different systems. Also a very highly engineered area, a lot of runoff going on there. It's all gravel and concrete and structures like that. But they're very water limited throughout their growing season, so that's kind of why they set it up like that. And you can see where, during the late part of the growing season, or into August, September, you can see where the scarcity starts to increase. You see the water footprint kind of spike in relation to the amount of water that they're applying or even kind of consuming. And again, the green water footprint at the bottom, it's applying, it's bringing in an amount of, but not a huge factor. You only see it really kind of change in what is essentially their off season from November to February, is when they get most of their rain up there anyway. This is kind of inland in the Pacific Northwest. And then we look at these two greenhouses, both at the same location. And the older greenhouse was about 1.5 acres in size, or 1 acre in production space. The newer greenhouse was about 1 acre production space, so they're kind of more efficient with their allocation of bench space and things like that, so they can grow plants. And it's overall larger. In this newer greenhouse, they use rainfall. They capture rainfall. The old greenhouse is essentially they irrigate out of a well, they apply overhead, and that's it. They get one bite of the apple. Whereas the new system is one that recaptures. They're using an ebb and flood system. So this is one where it kind of floods bench space, and then 80% of that is recovered back into the recycling system and then reapplied. And then they're also capturing rainfall. Otherwise, the production systems are the same for the kind of two different crops, two different tray crops. And I put a bold on this one here. You can't really see the light reddish, like the red hued ones are the old system, the blue hued ones are the new system. And the irrigation water applies essentially both for the same, same for both of them. But you can see how essentially having a water recycling system allows them to have a lower consumptive use. So they're getting more use to the water. And I mean, the climate characterization down there is sort of odd. It's generally water's kind of scarce, but it doesn't really spike at any point in the year. And then the last case study was looking at this nursery production system down in the Southwest. Once again, relatively large, 150 acres of production space, but the watershed is very large, almost 1,000 acres. And then I had to kind of fill in this because the overhead imagery didn't really show the channeling and kind of lower pond and upper pond system. They're essentially installing this new water recycling kind of system for their irrigation water. They're heavily dependent on water withdrawals from the Colorado River, and they have to pay for their water, so they have a really serious motivation to reduce their water consumption, or their water bill essentially. And you can see in kind of August, September, October, the scarcity really spikes there. And that is reflected in kind of this similar sort of graph. First off, there's no green water there. They're not getting rainfall when they're doing any production. Or, I mean, they do get some rainfall. We went through it, and the amount that they adjust their irrigation regime is basically non-existent for the amount of rainfall they get. I mean, they only get some rain even in December, and that's about it. But you can see how it drives up the water footprint during that time of high scarcity. And so to kind of look at them all together, you can't really compare these because there's a lot of confounding variables there. I mean, the length of their production system, how much they're pushing through. I think one of the things that would need to be investigated is to kind of bring in like a marketable net primary productivity of biomass that they're generating or something like that to kind of account for some of those differences in the production systems that are very much driven by the geography of where they are around the US and the water availability. But a few things to kind of note is that in all of them, the applied water is higher than the consumptive water use except for the Southwest. And when that first came up, I thought something was wrong. I mean, the Deep South doesn't really count because that number is the same. What's coming out of the well is being applied in the story. Everything else is lower except for the Southwest. And what is actually kind of going on there is they're making their water withdrawals to fill their water system during times of high water abundance, but so much is evaporating off, they're not even applying that much. But overall, it's for the amount of productivity they get out of it, you know, it's relatively efficient. But then when you look at it from a water footprint perspective, it's clearly the biggest impact for where they are. I mean, they are in the desert. It's kind of, I've been out there and it looks like Mars, and you know, bringing water on plants. And so that's kind of it here. Any questions? Dr. Coyne?
Not to be too critical of my colleagues on horticulture, but it strikes me that based on your presentation, that horticultural production is horribly extractive of water. And therefore,
Is there a question? So what is the question? If you could ask it again.
Am I interpreting your data wrong? I mean, that horticultural production practices that scale that you're looking at are horribly extractive of water, since you have such a little line.
Definitely that is the case for container production. Container production is much more dependent on irrigation. You know, these are essentially these soil media setups that they're highly drained. It's almost like sand, right? And so those systems are dependent on this constant, you know, introduction of water on kind of a scheduled sort of basis. And since that's not what the climate really provides, they're just more dependent on irrigation. Well, and I think that's why there was the funding for I mean, it was under a specialty crops research initiative to kind of look at water recycling and treatment and find ways to kind of quantify treatment and practice technologies based on their environmental impact. I mean, it's a problem. I think agriculture is 90% of global water use, just in agriculture. Yeah, horticulture's on for that. Go ahead.
Just give me any concerns about irrigation water quality. I know you're.
Yeah. Well, that's similarly what was kind of, we were just this sort of scoreboard piece of that, but you know, multi-state, a lot of KPIs, and yeah, like plant pathogens are probably the biggest concern, and they mostly look at treatment technologies to deal with that. I mean, that doesn't even include the weird xenobiotics and like plant growth regulators that people use or herbicides and trying to filter those things out so that we can water. That honestly is probably the biggest piece of the research, is making sure that you're not destroying your production system by reclaiming the water.
All right, let's thank our speaker one more time. our last speaker of the session is Mohammad Alsabri, and he will be talking to us about cell wall biology.
Hold the button down until the yellow dot shows up.
Point it more towards the screen, more on the circle. More towards like MS stuff.
Move the mouse. Oh, yeah.
Hi everybody, my name is Mohammad Alsabri. I'm a graduate student in the Plant and Soil Science Department. I'm here today to present my data, which is MS79, the microbially derived selective cellulose biosynthesis inhibitor. So the outline for this presentation, I will talk about briefly pesticides, and then what's herbicide, and then MS79 is our bacterial citrine that we are using to suppress plant growth, and then in the workflow, I will show you some picture and data from my result, and finally, we'll discuss about conclusion and future aspects. Introduction motivation. What are weeds? Unwanted plants growing alongside crops are known as weeds. Why are they a problem? They are fierce competitors, absorbing water and the nutrients with little remaining for the main crops. They grow really fast, creating shade around the plant, decreasing their ability to photosynthesize. Plants that lack water, nutrient, and sunlight will struggle, and ultimately the yield will be lower. In other words, our food will be lower. So what are pesticides? It's a synthetic chemical we use in agriculture to get rid of the pest problem. Herbicides, it's part of it, is one of the most important tools in modern agriculture to eliminate weeds. But weeds always develop resistance to herbicides. So for example, the United States of America is leading the world with 156 unique cases of herbicide-resistant weed. Australia in the second place with 84, and Canada in the third place with 64 unique cases of herbicide-resistant weed. There are also 36 weed species resistant specifically to glyphosate, and unfortunately, half of them here in the US. Therefore, we are searching for novel ways to attack these undesirable plant-threatening food supply. And for my dissertation research, I'm searching for novel herbicides from unexpected source, the microorganisms living within a plant. So when we look in order to figure out the solution for this problem, we look inside the plant, endophytic bacteria. These microbes, but they have to have a metabolic machinery in order to interact with their host. So it's an excellent source for producing bioactive chemicals. I'm going to introduce this class of herbicide in this slide because with the MS79, the ultimate chemicals that we use, it fall in this class. So let's talk about cellulose first and then CBI. Cellulose is a crystalline polysaccharide. It's important for expanding plant and growth, necessary for upright growth. That's why it makes it a suitable target for pre-emergent herbicide. So it's synthesized on the plasma membrane. There are some proteins are responsible for producing such as CSA. But CBI is a member of class L herbicide family and unique for the plant. And it is a very complex process. And since it's complex, it may hit the whole catalytic domain or the whole accessory protein, or even it will hit like the carbon flow in cellulose biosynthesis itself. So in the market, there is no mechanism of action identifying since like 20 or 25 years ago. We are trying to help to find a new mechanism of action to produce a new herbicide. So there are some widely known CBIs, such as isoxazoline, which is a synthetic one, and taxomin A. that is extracted from as a secondary metabolite for pathogenic bacteria called Streptomyces scapis. It's a plant pathogenic bacteria. So on the workflow, what we did here, we screened more than 200 bacterial strains, and we were testing their ability to suppress plant growth. And what we did is first, we examined radial swelling phenotype, and then I'll show you some picture about that. When we treated plant, sorghum specifically, our model plant with this bacteria, it will cause that swelling phenotype, swelling group. And then we did histochemical test for chlorobicinol. We put treated and untreated plant in a dye, then see the different coloration about the identification. And then we measure the cellulose content in the treated and controlled plant. And we realize there's a degree of selectivity when we treat with this bacteria. But the selectivity not between mono and diacot in general, it's just with the solanaceous. Solanaceous is family, specifically tobacco and tomato, is less effective. And then we identify the active ingredients in this bacteria by using X-algyricin and size exclusion chromatography. And then we decide to separate actual weed, not just model plant, Arabidopsis or sorghum, in the greenhouse and then in the farm. And then I'll talk about conclusion and the future aspect. So how does it work? I mean, NS79, the bacterial strain that we work with. So we inoculated the seed with this bacteria, and we got suppression of root growth with sorghum. And at the same time, this is how this bacteria looks like when it's grown in plastic tube or in MS media. This much of cells I will have from just over 90 gram of these bacteria. And it worked both ways. Inoculated the seed, in this assay will suppress plant growth, or using the metabolized of bacteria and then separate plant in the greenhouse, it will also suppressing plant growth. So the first graph, the first thing that we did is this axis is the root length, and this one is the overtime days. And these lines are representing the bacterial specimen, including MS79 and control. And you see here the root length gets really suppressed when we treat sorghum with MS79 compared to other lines that keep going to grow. So MS79 is suppressing root growth. So this picture is representing like the previous slide. We grow the bacteria until specific OD, optical density, and then we inoculate the sorghum seed. And then see here the clear picture about the treated and untreated plant. The treated sorghum seed has shorter roots than control. See the control? These are the control, and these are the treated. It's shorter and also it's swollen. So if you take close picture for this root, you will see the swelling sorghum root. It's a typical response when you add CBI such as antioxidant to the plant. So MS79 causes ectopic lignification in sorghum roots. We treated sorghum with MS79 and control without treatment. We put both of them in chloroglycinol dye. And I want you to focus on this picture on the red coloration in treated sorghum. This red coloration is came from like interacting between chloroglycinol dye and then the phenol ring in lignin. It's means like the cellulose was switched off and lignin was switched on. So our bacterial strain is causing like some dysfunction of cellulose in the roots of sorghum. We measure the cellulose, as I said in the workflow, and there's a clear difference between cellulose content in the sorghum that we treated with MS79 and control. So it's statistically significant. Selectivity. We test both, like dicot and monocot. 2 manic months I was testing, but I did not show like all of them. All monocot that we test in the basic assay, it gets suppressed. These are the treated, these are the control. It's very clear. The monocot with MS79 gets suppressed. But when we come to the dicot is a little bit different. There's, as I said, it's specificity with the solanaceous family. Here, tobacco and tomato, all of these are controlled. The up is treated. There is no big difference it gets some affection between tomato and tobacco, both treated and controlled, but not with Arabidopsis. Arabidopsis also gets suppressed, indicating a degree of selectivity. Here's a short movie about Arabidopsis seed. So this part is MS media supplemented with sonicated bacterial cells, and this is just MS media with the MSO. This is clear difference between treated and controlled. And MS-79 is suppressing the root growth. So we got the genome sequence for this bacteria, and it shows the recurrence pattern suggests it contains several plant microbe association clusters and cell wall carbohydrate binding and modifying gene consist with the bioactivity. Like isolation of active compound from MS-79. We did first XAD resin to extract the chemical, the metabolites from this bacteria first. And then we test this extract with Arabidopsis and sorghum in plate-based assay. We put control and treat supplemented media, and the plant gets suppressed. And then what we did is we did size exclusion chromatography on the same extract. So we put our product in a column, and then we mixed the growth that came from these columns with NS media to grow sorghum again on this extract. What we found is that the fraction that came at the very beginning, like which is 1, 2, 3, 4, 5, was not really suppressing plant growth. But the fraction that came up at the very end, 6, 7, 8, it's suppressing both Arabidopsis and sorghum. These are the controls. Indicated that our molecule is maybe a small molecule, maybe a protein or peptides, we don't know yet. We test the plant in the greenhouse. This is like a greenhouse experiment. I got some seeds, and then this is a valve leaves, and then this is Johnson grass. It's the same family like Arabidopsis, like sorghum, and then the sorghum plant. You see, I hope that picture is very clear. So these plants, they're treated with MS-79 sonicated bacterial cells, just very basic things. When I get a bacteria, centrifuge them, spray the cell from supernatant, and then sonicated the cells. The cell at 3 times for 5 seconds each one, and then spray the plant in the greenhouse in a pot. So these plants get suppressed compared to the control. See, it's very clear with these 3 plants. Another experiment in the farm, we got some spots in the south farm. These are the whole experiment. It's very simple. And then these, the picture representing the plant that not sprayed before spraying. These are closer picture. These after spraying. The picture is not really clear. The conclusion from this picture is if the plant is old, it will not affect it. If it's under stress, it will not affect it at all. But if the plant really small, it will affect it. These are black dots, it's dead plant. It's not really clear in the picture, but it's hard to catch them because once it's small, it will kill the plant. But if the bigger plant, it will not die at all. So in conclusion, our bacterial citrate compound is potential cellulose inhibitor. It shows greater effect on monocot than some diagot. And then the compounds produced by this bacteria are heat sensitive and small molecule. So for the future, we're gonna do more analysis such as like a GC analysis on NMR, running the HPLC, and measuring C14 incorporation to cellulose fraction of treated and untreated plants. Thanks for everybody helping me. My boss, Joseph, Dr. John, Dr. Khalid from Pharmacy School, Dr. Mark Williams, my co-advisor, Robert Janine, Tim Phillips, Luke Moon, Victoria, quickly, Steve in the South Farm, Sherry in the greenhouse, my sponsor at CED, my university back home, Wisconsin University in Babylon. And thank you for all of you for listening.
We have time for one question.
The red color you noticed.
Yes.
Sorghum. I mean, what do you think that is? Is that
It's lignification. The lignin starts to produce in this area, in this time in the root, but the cellulose was switched off. So because we measure the cellulose content in the next slide, if you realize, and then the cellulose content was less than and treated compared to the control. So the right correlation is lignification. Yeah, this for lignin.
okay?
It's very basic example. Just put the fluorouracil dye and treat it and untreated down. The control will not develop this color, but the treated will develop this color.
All right, let's thank our speaker one more time.